Good morning, good afternoon, and good evening. I am Viral Vigela, and I'm the account manager of X-ray CT technology with Wagaku. I'm here with our co-presenters, Ayat Takase and Angela Criswell.
Hi, everyone.
Hi, everyone.
Thank you all for attending Wagaku's webinar series, Ask the Expert. The best way to learn something new is talking to an expert. Our series will cover various topics and will be joined by industry experts to help us better understand CT techniques. Today, we want to welcome Josh Lomeo as the expert. Josh is the Director of Application Sciences at DigiM Solutions based in Woburn, Massachusetts. He'll be discussing how to advance in drug development with high-resolution X-ray CT. But before we start, a few housekeeping items. Today will be an interactive session, and I'll be sharing links as we go along the way. We'll be taking your questions live during the webcast and answering them after each question. As usual, please don't wait until the end to ask the questions. We won't be monitoring the raised hand function, and we'll be posting relevant links as we go along, so keep an eye out for them. We'll be answering as many questions as we can, and the questions we don't answer, we'll try to get to you by the end of the webinar. If for whatever reason you have difficulty viewing the webinar live, please note it is being recorded, and a recording will be available beginning tomorrow. With that being said, let me turn it over to Angela and Aya.
Thanks, Viral. Thanks, Aya, and thanks very much, Josh, especially to you. So I think we're ready to get started. So we're going to switch gears. Josh, are you ready to share slides?
Yes, I am. Let me get those up. All right. Can everyone see those okay?
Yes.
Yes. All right. So I'll just say quickly, thanks again. And we're going to dive in and ask Josh a few questions about drug development, how X-ray CT goes in. So first question is the obvious. What types of samples or products can be investigated using CT, Josh?
Yeah, that's a really excellent question and really key to designing our experiment and where we start thinking about applying this type of analysis. I want to first start off with where we typically don't apply CT analysis, and a common area that comes up is liquid suspensions. And one of the difficulties here is the length of the CT scan and the relative mobility of the sample. There's going to be Brownian motion, which can affect our scan as it's being performed, as well as the fact that we're oftentimes looking at features that are going to be sub-resolution. If we're thinking about the particles that are suspended or dissolved in the liquid media, they are oftentimes going to be at a smaller scale than we can see with even the most sophisticated CT technologies. Now, what can we analyze? Essentially any solid or semi-solid drug product or intermediate. So a couple examples we have here are tablets and capsules like Claritin, allergy pill, or any type of semi-solid cream or lotion like a cortisone can be analyzed as well. We can also look at small animal imaging. So this is an area that can be quite pertinent to understand distribution of drug within tissue, as well as the drug product. So if it's something like an implant or a microsphere, we want to actually evaluate where that drug product is and how it is degrading and evolving over time. Now, to look more closely at the types of solids and semi-solids that we can analyze, as I mentioned, one primary area is the oral solid dosage forms. So this can be any type of capsule, tablet, mini tablet, as well as all of the intermediates that the drug developers are using to manufacture these. So things like spray dried particles, granules, hot melt extrudates, these are all things that we can investigate with CT. We can also look at lyophilized materials. So this is something that often comes up during vaccine development when we need to freeze dry the sample for safer storage and stability. And CT technologies, especially with local tomography, are uniquely built to analyze these through the glass vial without removing the cake, allowing us to truly preserve the structure and analyze it without damaging it or exposing the cake to humidity and moisture. We can also look at devices and their powders. So inhalation is a common one, where we can actually look at the construction of the device, any defects or quality assessments on the overall construction, as well as the powders that are in the inhalation device. These can be studied with CT as well. And then in the category of kind of everything else, we can look at implants, microspheres, and ointments. These are some of the other less common dosage forms, but we do see them quite a bit, and great application of CT to likewise investigate these and some of the attributes important to the pharmaceutical scientists. So just to look at what a typical CT data looks like, this is what a typical tablet scan looks like, where we can actually visualize the distribution of active ingredients and inactive ingredients or excipients. And then we can also see within this tablet the distribution of porosity, cracks, as well as their different associations that they may have with each material phase. You can also take a look at an example microsphere scan. So here we can actually visualize hundreds to thousands of microspheres at once, assess their size, their shape, as well as things like their voids, if they have void spaces within them, and the encapsulation and uniformity of the ingredients within each microsphere. And with that, I'll take a pause and see if there's any questions on the types of samples that can be studied.
Yeah, question here is, what is the sample preparation like for soft samples like an ointment to be scanned on a CT instrument? Yeah, so the beauty of CT technology is that we can even scan the ointment in its inherent, in its tube. We want to try and avoid the often the aluminum tubing in the field of view, because that can introduce some hardening artifacts passing through that higher density material. But we can scan it in the tube, or we can simply squeeze out and dispense into a smaller plastic vial or tube. And that is a preferred method for many of our powder samples as well. Scanning in a plastic tube is a good way to go there as well.
All right.
Thank you.
Yep. So we'll move on to the next question. And what we want to know next is the sort of development challenges that can be addressed by CT.
Excellent question. Yeah, and we've seen quite a wide array in the pharma field, but we summarize it here in 4 primary areas. The first is process development. So when we're talking about manufacturing our tablet, as well as any intermediates, there are, of course, different manufacturing techniques within that process development, as well as the process parameters. So that can ultimately end up with a wide array of parameters to optimize in terms of how our structures are being affected in our drug product, and ultimately how those structures are affecting the performance. We can also look at formulation selection. So this is boiling down to which ingredients are we using, how much of each ingredient are we using, and CT technology is uniquely qualified to investigate all of that within 3D and in a non-destructive way. So this has been quite beneficial to pharmaceutical development to evaluate how the active ingredients and excipients are interacting, and weigh that on what should be going into that final tablet. We can also look at downstream performance. So this is coming up in oftentimes in the area of troubleshooting when we don't understand why a certain tablet is dissolving or disintegrating faster than another tablet. This can be within a batch, even the same manufacturing batch that we see variations, or across different batches. And then in the fourth area here, we have CMC and stability. So CMC is chemistry, manufacturing, controls. This is primarily associated with the safety, quality, and efficacy of the drug product, especially as we are doing things like technology transfer, scale up. We want to ensure that the quality of the drug product is maintained as we are changing sites or changing equipment. And then the other component here is stability. So oftentimes as part of ICH studies and part of the regulatory submissions, we'll need the pharmaceutical scientists will need to expose the drug product to high humidity, high temperature conditions, and evaluate how that affects performance. And that is going to ultimately guide things like the packaging selection, the overall shelf life, and the idea of the expiration dates on the packaging. These are all things that the stability studies go into. And I will mention that many of these challenges areas are applicable to other industries, from batteries to filters. And we do have a fun example here, actually looking at a little snack break here, where we can look at the importance in potato chips, where we have done a CT scan. So in this instance, we're looking at a Lay's potato chip, and we can think about the process development, how we are making the chip, the formulation selection. So here it's what is the amount of salt, the amount of oil, and the amount of air. And then when we think about downstream performance, this is, in the case of a potato chip, really the consumer experience. So what is the overall taste of the potato chip? What is the mouthfeel? It's a very food science oriented goal there, to have a good mouthfeel based on whatever product we're looking at. What is the smell? And then kind of on the stability side again, what is the shelf life and the bowl life? Bowl life is actually quite an interesting parameter that we may have all experienced when eating our cereal. How quickly does the cereal take up the milk and become soft is a specific parameter that food scientists look at. So this is all to say again that CT usage in this R&D space is certainly not exclusive to pharma, and there is even applications in things like food science that we've seen here. And that I'll see if there's any questions on this area.
Question from, I guess, when we were transitioning to the second question, somebody asked, how do you measure diameter of microspheres? And I know there's multiple techniques, and we briefly touched on light scattering, dynamic light scattering, but in terms of just the two techniques, optical imaging or light scattering and CT, you want to comment on that? And then the other question was just briefly, when do we want to start using X-ray CT in development process?
Mm-hmm. Yeah, so I think we'll probably touch on that second one in a moment. But with regards to the first question on size calculation, so we are using our machine learning analysis algorithms to classify each microsphere and differentiate it from the air. And then once we have that classification of each microsphere in the scan, we'll then typically measure with a equivalent spherical diameter algorithm. There are, of course, other algorithms that we can incorporate and apply the size measurement with, but that's our go-to measurement technique there.
Yeah, definitely. Yeah, so that goes back to segmentation and things like that we've covered in previous webinars. So once you assign them, you can certainly do all sorts of quantitative analyses. I love the Lay's potato chip, so I'm wondering what that chip would look like if you left it in a bowl in the air for a few hours. You and the test.
Yep, indeed.
All right.
They
so I
actually
guess we-
stay pretty crispy for, I don't know, unnaturally long time.
Naturally they do, don't they? That's yeah, very true. So okay, let's see what our next question for Josh is. It's a question for the audience.
Yes. Okay, so we're gonna have a couple questions for the audience throughout the webinar, and here is the first one. So let me launch the first polling question. So the question for you is, do you often use the imaging in your projects? You're either research and development or process control projects. And you can choose from yes, sometimes, or no. I think we're seeing a lot of it yes and sometimes.
Yes.
Good to
Answers
hear.
are coming in pretty fast. I'm gonna give you a few more seconds. And we're gonna close the polling in 3, 2, 1. Okay, let's take a look at the results. So 57%, almost 60% of you said it yes, we use imaging, and 33% sometimes, and 10% no. Good to know that the
Alright.
most of you use it.
Yeah. So this is absolutely relevant to some of the work you guys are already doing then, for sure, for most of you.
Okay, let me stop sharing. There you go.
All right. Thanks for answering that, everyone. We'll go to the next question for Josh now. All right. So in this question, Josh, what does the typical design of an experiment look like when we're using CT data to support development?
Yeah, on that front, one of the first things we look to is what exactly are we looking to quantify? And that's a really important goal because we want to, really understand how we're designing our experiment around those objectives. And this can come in drug development to a variety of things like, do we want to understand how the ingredients are interacting within the tablet or in the drug product? The distribution of the ingredients. So do we want to understand if everything is uniform across the drug product or if there's perhaps some heterogeneities? The internal particle size is oftentimes a measurement we're making. The assessment of porosity, voids, fractures often comes up. Particle morphology is another area that can be either within the drug product or within a powdered intermediate, like a spray-dried particle or a granule. And then uniformity within and across batches. So this can be something where we actually need to consider scaling our CT resolution back. So rather than scanning one tablet, we can scan 3+ tablets in one scan to assess their overall uniformity and consistency of structures that we may have observed at higher resolution. So some of the other typical questions, what is the sample size and region of interest? And that's going to ultimately determine our field of view. So we want to scan the whole tablet. Are we aiming to scan in a region within the tablet? These are key questions that we ask our clients as they're coming to us with their development challenges. And we go about designing these experiments. A related parameter here is what is the feature size? So of course, if we can go in knowing what the size of our particles are or what the size of our pores are, then we have an idea of our needed resolution for the CT scan. And typically we use a rule of thumb there of having 3 to 4 pixels to have confidence in analyzing and quantifying the feature. So that's a good guide there to make sure that our resolution is 3 to 4 times smaller than that feature size, so we have good confidence in resolving it. And then the last component here is what are the ingredients in the tablet or drug product and what are their relative densities. So this is going to guide our expected contrast that we see within the CT scan. So we're looking at primarily the difference in density as well as higher atomic Z numbers. And going into our scan, if we know what the materials are and their relative densities, we can have a really good expectation of which phase in the scan is corresponding to which ingredient, and then give us confidence as we go towards analysis and quantification, knowing which domains that we are looking at. And with that, I'll stop, see if there are any questions on the experiment design for CT scans.
Let's take a look. Is there a guideline in defining a field of view to make sure that you have a sound representation of your sample?
Yeah, so typically if we're talking about, I'll use a tablet as an example, the best field of view there is ultimately the full tablet. And as we do that scan, it does allow us to see if we're getting some sense of features that we may see better with higher resolution. That'll help us pinpoint where within the tablet we need to make that scan. There is, of course, the question of representativeness as a whole. This is only one tablet, only one sample. How do we make sure this is representative? And that's where we would go about scanning multiple tablets at a wider field of view to ensure that they are looking similar across the board.
Yep, makes sense for sure. All right, so that's a good guideline for the experimental design here. Thanks so much, Josh. So if we move on to our next slide, this is a great one. At what stage should you start considering CT in your development process?
Yeah, so we've seen that ultimately the earlier the better. So there's really no too early a stage here, but we'll break it down into some of the core areas here that we've seen and what the primary studies are for each of those. So looking at preclinical stage, this is going to be primarily focused on the how the active ingredient mechanical properties are, as well as the interaction with excipients. So this is going to help us understand as we're granulating materials, as we're tableting and compacting them together, how are they interacting with the active ingredient? And that's a really key fundamental property to understand the physicochemical properties of that active ingredient. A next step up from that is ultimately going to be the formulation, selection, and decision making. So once we start to choose our formulation and our different ingredients and the amount of each, we want to connect those structures to release behavior and key in on the formulation that gives us the most optimal performance based on the intended use. So that can be something like an immediate release, or it can be something more extended like a controlled release. And looking at this CT analysis can be a really key area to key in on those best formulation lists. You can also look at the clinical phase. So this is where during drug development, we really start to key in on optimizing manufacturing techniques and process parameters. And an additional part of this, as we are moving further downstream and through the clinical phases, we are going to have scale up. So going from lab to GMP scale, and ultimately even larger manufacturing sites, we want to ensure that we're reducing performance variability and any issues that may come up during the scale up. In terms of post-approval, common area here is SUPAC. So this is a regulatory component where we may change manufacturing process or site, and we need to ensure that the drug product quality and efficacy is equivalent to our previous manufacturing process and previous sites. So this is a way where CT analysis can prove quite valuable to demonstrate that structural equivalence between old and new locations, as an example. And in general, enhancing future developments through a knowledge database. So the knowledge gained from the CT analysis does not need to be excluded to that specific drug product. We can begin to apply our understandings and our learnings from that analysis to similar products which have are either similar in how they're manufactured or are using a similar set of ingredients and excipients. And with that, yeah, are there any questions on stages of development to use CT?
I think that was a previous question from a couple of questions ago that you just answered, but just curious, how does CT complement other analytical techniques that are used in various developmental stages?
Yeah, overall, it's quite orthogonal. I would say that there are going to be always those more routine measurements that are based on their speed that will more often take. But the beauty of CT comes in the ability to really get a three-dimensional and non-invasive inspection of these. So we talk about things like particle size, porosity. There are, of course, complementary techniques like laser diffraction, mercury porosimetry, pyknometry. We recognize in the CT world that these are always going to be perpendicular techniques. But with CT, we do have that added benefit of being able to quantify and assess many of these parameters in a single experiment and really get a finer detail understanding of them that we may not be able to otherwise.
Yeah, and I can imagine, in terms of delivery systems, or if you're using a device, this stuff could be pretty powerful both before and after approval processes. So yeah, it makes complete sense, Josh. So if no more questions, let's move on to our next question here. And surprise, it's another one for the audience.
Okay. So this is the second question for the audience. So looks like about 2/3 of you said that you do use on imaging analysis. Now the question is, do you analyze the images yourself? If so, you can choose a yes. Maybe sometimes you do it yourself, or you might say, no, I ask somebody else to do it. So who does the analysis of your images? Looks like most of people say yes, they do the analysis themselves. Okay, so most of you, 65% of you answered. I'm going to give you a few more seconds. 3, 2, 1. And I'm going to end the poll. Oh, I got a couple more votes in the last seconds. And I'm going to share the results. So 64% of you said yes, you do it yourself, and 23% sometimes, and 14% of the audience said, no, I ask somebody else to do it.
Excellent.
Okay, thanks for answering. I'm going to now stop sharing.
All righty, and that'll take us on to our next question. And in this question, Josh, we want to get at, you to tell us how CT data might support regulatory filing. Can it, and if so, how?
Yeah, absolutely. And that's quite a common application that we see inspecting some of the things that we've talked about. So in general, when it comes to the FDA and regulatory agencies, we want to common application here is the visualization and quantification of drug domain size within the drug product. This is something that the CT technologies are very uniquely qualified for, as opposed to other techniques where we may need to, say, cut open the tablet, cut open the capsule. Here we can do all of this measurement non-destructively within the final dosage form. And that is frequently a request that we see from the agency. And this can be expanded into a variety of other regulatory requests on physical chemical properties, such as porosity, that we may want to evaluate within the final drug product. Again, the ability of CT to do this non-destructively and in 3D is really makes it stand out amongst analytical techniques and amongst microscopy techniques as well. For generic developers, one of the other applications that we see is the demonstration of structural sameness with the reference listed drug. So this is an important area as they are developing their generic product to demonstrate that sameness with the RLD, as they call it. And this can even be expanded a little bit further to Q3 bioequivalence. And so this is applicable only to a smaller set of generic products, primarily semi-solids, so topical lotions and creams. But in this instance, they can actually use that structural sameness through a Q3 bioequivalence demonstration. They can use that as a bio waiver for further testing. And this is also a larger part of the, this is essentially the third step in the Q1 and Q2 equivalents where they need to demonstrate the sameness in ingredients that are being used. Yeah, any questions on the use for regulatory filing?
I guess what software can we use to provide CT data and analysis in the CFR Part 11 environment?
Yeah, on the software side, so we've actually designed DigiM I2S cloud-based platform to manage that compliance. And that the platform is actually built around the idea of better data management and visualization. And on the regulatory side and adhering to that compliance, allowing a pathway for easy data auditing. So this will be a way where we can track what has been done with data, who has done that type of, who has done the analysis, when they did that analysis, both for the original, tracking everything for the original CT data, as well as any derived data sets that might have been extracted during the analysis.
Yes, that's you guys have an excellent tool with your I2S system there. So I imagine they want to see exactly what happened, when it happened, and the result of it. So it's key to have all that stuff. All right, so that'll take us to our next question here. And surprise, Josh, we have a few questions for you that are not related to drug development. We call this our lightning round. So we're just going to ask you a few questions. Maybe we'll get to know Josh a little bit better. So question one, Josh, where'd you go to school?
I went to the University of Rochester in Rochester, New York.
Excellent. And a trick question, it's not really a trick question, but what's your favorite imaging technique?
I have to go with micro CT. I love the dimensionality of it, and super broad number of applications that we can see applied across different industries.
Excellent. And so in terms of drug development, we don't see a lot of integration of CT. Do you think CT will be utilized more or less, or will it be utilized differently? What are your thoughts on that?
Yeah, I think we can only go upwards as the industrial developers are gaining awareness of the technique and how it's being applied in the process, as well as agency's knowledge of this type of technique. In their guidances and recommendations about different ways to measure and measure attributes, that's only going to gain ground and further interest within the agencies, and ultimately our hope is ultimately in the guidances to the developers to use that type of technique as well.
Excellent. And I guess we'll find out in the future. I'm looking forward to what CT can do rather. So one other question: what would you be doing if you hadn't have gone into your current field?
Yeah, so I have an interest in antiques and collectibles. So my other dream job would be getting involved in auctioneering and selling antiques. So
that's pretty cool. And you know what? You could probably analyze a lot of those things with CT as well.
You sure could, yeah.
Yeah, definitely. Cool. And then we're going to finish with one last question. So what's your favorite science podcast or science show?
I would have to go back to an old favorite in Mythbusters. Yeah, they always kept science very interesting and fun, and yeah, that would have to be my favorite there.
That's a good one for sure. It's a great show. All right, so enough of that fun. We're going to go back to some science now and ask Josh another question. Surprise. It's for you guys.
Again,
the audience.
it's the audience's turn. Okay.
There you go.
Right. So the third question we have for you today is this. Is data management and sharing a challenge for you? So we've been talking about image analysis, but now we're talking about the data management, including storage and sharing.
I was just tackling some of that this week, right? So moving things,
Yes,
backing up, yeah. Archiving.
because it's three-dimensional, the CT image files can be huge.
Yes, yeah, for sure.
You can run the scans casually, then you realize how much of the data you have on the drive.
Exactly. Definitely got to stay on top of it.
Yep. Okay, so most of you answered the question. I'm going to give you a few more seconds. Now in 3, 2, 1, I'm going to end the polling and share the results. Okay. So more than a half, about 60% of you said yes, you find it challenging, and 35% said it's somewhat challenging, and 6% of you figured it out. It's not a challenge.
Can we talk later?
Yeah. Okay, I'm gonna stop sharing. And back to you guys.
All right, so let's head on to the next slide and see what's next. All right, so the question here, Josh, is we want to know, how does CT data and simulation support dissolution analyses?
Yeah, that's a really great question, and where a majority of our pharmaceutical clients are coming to us with their challenges. I want to actually answer this through a couple case studies that we have with clients on a couple different types of drug product types as well. So this first project was a publication with Eli Lilly, where we were looking at dissolution troubleshooting upon scale up. So going from R&D to GMP batches and understanding the root cause differences between those performances. So this is a paper published in the European Journal of Pharmaceutical Sciences. And in this project, we were looking at lab and GMP batches, in this case of a spray-dried particle powder corresponding to P1 and P2, P1 being from lab, and then P2 being from GMP. And these powders are ultimately compacted with excipients to form respective tablets, their respective tablets T1 and T2. And where the Lilly team was seeing a challenge here is the relative dissolution rate of each tablet. So in the lab batch T1, we see really nice immediate release dissolution. We're about 100%, nearing 100% release at about 20 minutes. Whereas in T2, the GMP batches, we're looking at up to about 80 minutes, and we're only at 80% release. So quite a drastic difference in their performance. And where CT data came in here was actually looking at the corresponding particle batches and their tablet batches. So P1, P2, and then T1 and T2, where we've scanned both the powders and their corresponding tablets. And one of the areas on the powder front where SEM and all the other physicochemical techniques kind of failed in seeing any differences, with the XRCT, we could actually see morphologically differences in individual particle behavior, as well as the relative clustering of the particles. So we see, for example, in P2, we have this large kind of honeycomb structure that is revealed by the CT scan, which would have been difficult, if not impossible, to see with surface SEM or any other surface microscopy. Even further than that, we can actually extract quantitative differences in things like the relative particle size, the wall thickness of these spray-dried particles, as well as their exterior surface area. And these are all things that are going to be critical as we take these powders and we compact them into a tablet. And how they're going to compact is ultimately going to affect the porosity within the tablet, and then that will lead to differences in dissolution and disintegration. And so that's where our tablet data comes in here. So from the tablet CT scans, we actually quantified the connected porosity within each tablet. These two visualizations in blue are all of the connected pores within each tablet. And we see that T1 has a very nicely connected porosity regime. And then in T2, we actually have some large clusters of disconnected porosity circled in green here. And this is ultimately some of the key findings from the study. And we were ultimately able to tie that back to the dissolution performance. So as you recall, if you recall, the lab batch, we have nice immediate release, whereas the GMP batch is taking quite a bit longer, over an hour to dissolve and release the drug. And so this ultimately tied back to looking at the tablets, the porosity within the tablet, as well as the particles that created that porosity. And coming back to the complementary, the orthogonal techniques, this is where CT is really unique in being able to see not only the porosity, but how that porosity is connected amongst itself. And CT analysis is really uniquely qualified to make that assessment. Another area that we often support dissolution analysis is with controlled release platforms, so implants, devices, microspheres. And in this instance, with Innocor, this is a project funded by the Bill and Melinda Gates Foundation for a bioresorbable injectable contraceptive implant. And our aim here was to really reduce the need for long in vitro testing, understanding the release mechanism and behavior of a novel polymer that Innocor has developed, and being able to correlate in vitro and in vivo release performances. So one of our initial investigations in this project was actually looking at the mechanism of release and how our release profile can be understood through CT imaging of the implant over time. So what we ultimately did here was uncovered through the analysis of different time points how the implant is degrading how the polymer is degrading, and what that means for the overall dimension and mechanical properties of the implant. So as we look along here from left to right, we see how the drug release front is penetrating from the outside to the inside of the implant. And then at later time points, we actually see how that layer is peeling off. So that's going to actually give us essentially a new surface area to dissolve the drug from inside out. And then at our later time point here, we even see some cracks forming within the implant. And then this is something that in the world of drug release is going to give us more dissolution media penetration and ultimately a acceleration in drug release. So all in all, this time-based imaging, collecting different implants from different release testing times, allowed us to actually understand mechanistically the accelerations in drug release over time for this implant. Now we can also look at this degradation and evolution both in vitro and in vivo. So we have the comparison with 56 days in vitro and in vivo. We see that the in vivo core is holding up a little bit stronger, where we have a diameter of 1300 micron, as opposed to the in vitro, which is at about 1200 micron. We can look at even later time points in the case of the in vivo. At this point, the in vitro has completely released and degraded, but our in vivo sample is still releasing at this point. And here we can again assess the structural evolution of the polymer as it's degrading. In this case, we're down to around 700 micron. And here we can even visualize at the leftmost portion of the yellow circle, we can see a little bit of lingering tissue on the implant as well. We can even go a step further here and simulate drug release, in this case, from this implant. And this is something where we are actually using the structures that we've visualized with CT, quantifying those structural networks, and then using that to predict release. And this is really powerful because we're, at this point, reducing as much as we can any assumptions on what those structures are looking like, because we've actually visualized and quantified those particle networks across the device. And this has been quite powerful for in the controlled release and long-acting space for our clients. And just an example of what that data looks like here. So we have a comparison between in vitro, a couple in vivo curves, as well as our simulation. We see that our simulation is agreeing quite well with the in vivo time points, especially at later time points. And it does allow us to actually continue to project and see what the expected duration of release is beyond the in vivo. And so this, the CT collection and the simulation prediction and all the analysis that goes into that can be performed in a matter of a few weeks, as opposed to, in the case of this long-acting implant, where we're looking at, in some cases, years-long release testing that needs to be evaluated. And this can ultimately save a lot of time in vitro and in vivo testing, where the drug developers are making a drug product with different formulation, different process design space, and then doing the testing to see how it performs. With the image-based simulation, we can have a projection of what that will look like from the time zero sample, and then that can save a lot of time in that formulation and process development. Yeah, with that, I'll take a pause and see any questions on dissolution performance assessment.
I don't have a question, but I'll ask one. It was really interesting when you were mentioning the case with Eli Lilly and the poor connectivity on T1 and T2. Do you think that could have been solved with without X-ray CT usage?
Yeah, jump back here. So there may be other pathways to, get to this point, but I think it would have taken a significantly longer amount of time to reach this conclusion. And this is kind of coming back to the question on orthogonal techniques. It doesn't exclude the possibility of reaching this conclusion with them, but it would have taken quite a bit longer, say with something like mercury porosymmetry or picnometry, to understand that the porosity systems were this different in their actual structural interaction. I think that assessment would have taken quite a bit longer with more traditional lab techniques.
Thank you. And I think we had one more question in the chat was, can holograms be used to recreate CT images? I don't know if that's something you've come across or thought about, but
Holograms, is that?
yeah.
Yeah, I think there's also a reference to, do you print these as well?
Okay. Yeah.
Basically getting it to the point you can see it in a larger scale, I guess.
Yeah, there's definitely different applications there of digital transformation, and even 3D printing some of these scans and analyses can be really useful. There's definitely a space there to look at imaging data, both artificial and generated, ways that we can generate structures and analyze their properties is definitely something to look forward to in the future, and can be supported by the CT analysis.
Definitely. I think the best, one of the best educational example I've seen recently in terms of 3D printing is a guest who had a talk at a recent conference I was at. It was actually the Dragonfly user meeting. They 3D printed snake fang and illustrated the venom, how it comes out of the fang, and they use this at the museum to sort of teach the visitors how that process works. It's really cool. Sometimes seeing how things work is really neat. So yeah, it was really, really cool. And also I love the fact that you guys are doing this simulation, because we're seeing more and more use of data like this to do simulations, whether in the drug development. We see it in simulating how batteries work, how they discharge, filtration, digital rocks, all of these things. And I think that they have the potential to really utilize this data in a way that we would take forever to get to some answers. So so for sure, very powerful stuff.
I actually have a question related to the simulation, because we got I have a few more minutes.
Yeah.
So Josh, do you think people will be using simulations to replace more expensive and time-consuming experiments, or I guess you can use the simulations to simulate different scenarios in terms of changes you can make to the production to address certain issues, instead of trying one thing and running 1 year experiment and trying another thing and running another experiment. So it could be both, but where do you think simulations can be utilized the best?
Yeah, so I think there's already a good place for this in the R&D space. I think the next step will be acceptance by the regulatory agencies as for this more in silico prediction in the regulatory filings, to either support or in some cases actually potentially replace going into those deeper measurements with, say, in vitro or in vivo data. There's always going to be that need for the in vitro and in vivo data, but one of our ultimate goals is to drive home the point that this can be used to support and complement and perhaps reduce the amount of resources that needs to go into that collection.
That
I
makes
see
sense.
a lot of opportunity there for sure.
Thank you.
All right. So let's go on to
One more.
next
This is the
one.
last polling question.
Yes.
Okay. So here is the last question we have for the audience. So what kind of images do you collect? We've been talking about just X-ray CT, but that's not by any means the only imaging techniques. Okay, so answers are coming in very fast. You can check as many as you want.
We were quite curious to see how this one shapes up.
Yeah.
Me too. Yeah, for sure.
Looks like almost everybody uses something. I'm gonna wait on a few more seconds. I guess some people are still thinking and trying to remember what they have in the lab.
Yeah, for sure.
Okay, so I think a majority of you made your choices. So I'm gonna end the polling. And let's take a look at the results. Okay. So I guess SEM and light optical microscopy they're tied. They're the winners. And micro CT comes second. Then TEM, other techniques, and some cryo-TEM users.
Very interesting.
Yeah, it's a big spread, isn't it?
Not surprising, though,
Mm, mm,
I'm gonna say. And of course, we want to see more micro CT, but
yeah.
Yeah,
I see a
recognize
lot
there's
of SEM.
always
Yeah.
a time and place for each of these, right? Depending on what you're
Right.
studying.
And the
and
questions
they
you have,
complement each
yeah.
other. Yeah.
Yeah,
All
for
right.
sure.
Well, thank you for answering this question too. And I'm gonna stop sharing. Go.
All right. Thanks so much for answering that question, guys. It's pretty interesting to see all those answers. And we're going to go to the next slide. And Josh is going to kind of wrap it up and tell us what DigiM does.
Great, thank you, Angela. Yeah, so DigiM, we are a technology leader for characterization-based solutions, primarily on the microstructure scale, but really any type of analytical problem that can be solved with micro CT or imaging analysis. So we've seen a really growing interest in the use of 3D imaging data, reflected most by our gray curve here, where the unit on the y-axis is 50,000. So we're looking at over 4 million images that we have analyzed in-house. So micro CT and 3D imaging data is really proving powerful, especially in the area of drug development, where it's gaining good ground. In terms of DigiM, we provide contract research to transform product development, from the imaging collection all the way up to the AI analysis and image-based modeling. This can be either from your imaging data or images that we collect. We do also have a software platform, DigiM I2S, and this is a pathway for image analysis, prediction, and data management, all in one in a cloud-based platform, to really make sure we're making the most use of our micro CT data and extracting meaningful quantitative data from it. And with that, I will pass it back over to the Rigaku team.
Well, I'd just like to take this moment and thank everybody for attending today's session. As suggested earlier, the recording of this webinar will be available tomorrow. And join us next month. We will be chatting with Dr. Arne Jacob from Map to Market, and we're going to be discussing digital rock physics on May 17, 2023. Until then, take care and keep scanning.
Thanks.
Thank you.
Bye, everyone.
Bye.